see. Dark candy better here to play. If you don't come play with us, then we will kill you.
Diabetes mellitus is a chronic, lifelong condition that affects your body ability to use the energy found in food. There are three major types of diabetes, which is type 1 diabetes, type 2 diabetes, and gestational diabetes. Normally, your body break down the sugar and carbohydrate that you eat into a special sugar called glucose. So, the glucose will fill into the cell, but it needs help of insulin. Insulin is a hormone in our bloodstream in order to take in the glucose and use it for energy. With diabetes mellitus, either your body doesn't make enough insulin, it can't use the insulin it does produce, or a combination of both.
Glycolysis process begin with molecule glucose. The enzyme that begin the process is hexokinase or glucokinase in the liver, and ATP donate phosphate to glucose, which then form glucose 6-phosphate. Next step is glucose 6-phosphate is rearranged into fructose 6-phosphate by using enzyme phosphoglucose isomerase. Isomerase are enzymes that rearrange structure of substrate without changing the molecular formula. Now we made another kinase, that is phosphofructose kinase. This enzyme attach another phosphate group that are donated by ATP to a molecule, which creating fructose 1,6-biphosphate. Next, the molecule is splitting half by fructose biphosphate aldolase. Fructose 1,6-biphosphate is split into dihydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate or G3P. Next step is converting the dihydroxyacetone phosphate DHAP into glyceraldehyde 3-phosphate or G3P by using enzyme triose phosphate isomerase. Next enzyme we made is glyceraldehyde 3-phosphate dehydrogenase. In this step, 2-NADH is produced. There are two molecule NADH because one molecule glucose create two glyceraldehyde 3-phosphate, which is one from aldolase reaction and the other one is from triose phosphate isomerase reaction. The hydride group in a molecule is replaced by phosphate supplied by inorganic phosphate. The reduction of NAD plus to NADH provide energy to bind inorganic phosphate. This create a molecule 1,3-biphosphoglycerate. Here, the molecule made another kinase, that is phosphoglycerate kinase. 2 ATP produced by taking phosphate from 1,3-biphosphoglycerate molecule. This produces a molecule 3-phosphoglycerate. The enzyme phosphoglycerate mutase create an isomer 2-phosphoglycerate from 3-phosphoglycerate. Enolase is a next enzyme and it produces a double bond by removing its hydroxy group. This creates phosphoenol pyruvate or PEP. The final enzyme is in glycolysis is pyruvate kinase. This kinase also removing phosphate from molecule attached to an ADP, which then create an ATP. Again, 2 ATP created because 2 PEP will be produced for each glucose. This finally lead to a molecule pyruvate, which then will be substrated in TCA cycle. Overall, there are 10 steps in glycolysis, which involve 10 enzymes.
Now, let's move on to the next topic on carbohydrate metabolism, which is gluconeogenesis. First of all, we're going to start with 3 carbon molecules of pyruvate that can be the precursor to form oxaloacetate by using pyruvate carboxylase, where ATP is being used and reduced to become ADP. Oxaloacetate is then being decarboxylated and phosphorylated to form phosphoenol pyruvate by using enzyme PEP carboxylase. During this reaction, G3P is being hydrolyzed to form GDP plus carbon dioxide. Next, phosphoenol pyruvate will use N-nolase and water is being added in this reaction to form 2-phosphoglycerate. Next, phosphoglycerate mutase is being used by 2-phosphoglycerate to transfer phosphate group from carbon 2 to carbon 3 to form 3-phosphoglycerate. Phosphoglycerate will then use phosphoglycerate kinase and reduce ATP to ADP to form 1,3-biphosphoglycerate. From there, we're going to use glyceraldehyde 3-phosphate dehydrogenase, where NADH will become NAD plus inorganic phosphate to form glyceraldehyde 3-phosphate or G3P. On the next step, the aldolase is being used to form 6 carbon molecules of fructose 1,6-bisphosphate. Then, fructose 1,6-bisphosphatase is being used by fructose 1,6-bisphosphate to become fructose 6-phosphate. Then, fructose 6-phosphate will be converted to glucose 6-phosphate by using phosphoglucose isomerase. Then, by using glucose 6-phosphatase, glucose will be formed from glucose 6-phosphate. That's all from me for gluconeogenesis. Thank you.